Welcome back to another session, the sixth video of DNA rejuvenating technology. All the technology explained at your grasp, at your service. And today's topic is large scale expression of AAV-OSK virus in HEK293. The mammalian cells, this kind of specific cells that we are going to use. And what does it mean? It means inserting OSK DNA, RepCap DNA, and helper DNA inside the mammalian cells, the HEK293, producing millions of copies of the OSK genes and encapsulating the copies inside the EV particles using this kind of cells that I have told you. Okay, let's have a look here. We have the mammalian cell here, and what we are going to do is co-transfect. That means include the three DNAs inside the mammalian cells. The OSK DNA, the RepCap DNA, and the helper DNA. And the cells will do all the work for us. The cells with the DNA will produce many copies of the OSK genes. You see here the copies of the OSK genes. The OSK genes, remember, are where in the OSK DNA. Then the RepCap DNA will serve the following purpose. It will produce EV particles that you have here. All right, and the helper DNA will help the RepCap particles to include the OSK genes. So the whole process will be producing many copies of the OSK genes and include the copies inside the EV particles. And afterwards, the cells will secrete many millions of EV particles, EV virus with the OSK genes inside. Easy, right? Let's go for it. Okay, we are going to start today's video, the sixth one, large scale expression of AAV-OSK in HEK293. And before starting, remember, we need a reminder, a little bit of context of what we are doing, okay? The therapy and a little bit the process. Let's go for it. Let's go for an introduction and afterwards for today's topic. Let's go, let's start. Well, let's remember in a couple of minutes what we learned in the first video. We learned about the DNA, this beautiful molecule inside our cells. It contains information to shape our bodies, to build everything. Your cells read the DNA as a manual to build all their components. Okay, just remember, the DNA is just information. It's a guide to build things in our bodies. Okay, after this reminder, let me ask, do you remember something else? Maybe these little marks in the DNA? I show you that the DNA is modified with these little marks that you have here. Have a look. You see the little marks, right? The marks are stamps. They provide extra information. And sometimes they say, this DNA is old. Don't read it. It's a shame when you read it. Don't you think? When our cells see the marks, they literally read, this DNA is old. Don't read it. Well, when this happens, our cells lose functions. They get older. That is correct. As time passes, the amount of marks increase. Do you see here? This DNA contains lots of marks. As time passes, the DNA piles up a lot of these tiny marks. Well, in this DNA, the one with lots of marks, the information is no longer available. Is no longer read, and our cells get older because of that. as a consequence of the marks in the DNA. That is correct. Our cells stop reading parts of our DNA. Well, it's when you get older. I'm sorry, my friend, but don't worry. Our cells have a solution for that. It's called OSK, a magic potion to revert the age of the DNA. OSK clean many of these marks. OSK polish the DNA. It reduces the amount of marks and the cells are again able to read the DNA. In other words, we rejuvenate. Oh yeah, we rejuvenate thanks to OSK. Great, right? Good to know. Well, that is the key of this technology. We provide OSK to the cells to rejuvenate them. We reactivate somehow OSK and young again. OSK, young and healthy again. Cheers! Okay, let's remember how to produce OSK. Let's go for the next slide. Before starting with today's session, let's remember the overall process. Let's remember how this technology works, how we produce the EV particles with the DNA inside, with the OSK genes inside. Well, remember, to we had to design good DNA, okay, with the OSK genes. This is the DNA. We had to design good DNA with the RepCap genes. These are in charge of producing the EV particles and the helper DNA. This DNA will help the process. That's all. So in the first step, we introduce the DNA inside bacteria cells. And the bacteria cells are very clever. And as we grow the bacteria cells in culture, they will produce many, many copies of the DNA, as you can see here. Once they produce a lot of DNA, we extract the DNA. And yeah, let's go for the second step. And the second step is today's topic. So the DNA with the OSK genes, is expanded and encapsulated inside EV particles. This is what happened in the second step. So once we extract the DNA from the bacteria, we introduce this DNA. We need to introduce the three DNAs inside one cell because they work together. They coordinate themselves to work together. So we introduce the three DNAs inside a mammalian cell called HEK293, and this cell is super clever. This cell with this DNA inside will be able to produce many, many copies of the OSK genes and encapsulate them inside EV particles. And the third and last step will be purifying, cleaning these EV particles, leave them ready to work, leave them ready for delivery, for helping us to keep ourselves young, healthy again. So let's go for it. Let's go for today's topic. Okay, let's go for today's topic. DNA with OSK expanded and encapsulated inside these cells. Let's go for it. Well, we are in the summary slide. This slide summary the 5 steps involved in producing EV-OSK inside mammalian cells, which is a process that is called large-scale expression of EV-OSK inside mammalian cells. And what is the first step of the process? Well, the first step is just preparing the mammalian culture. So the day before, we prepare the culture of mammalian cells that are called HEK293. And because we will need 10 liters of culture for this purpose. What are the parameters of the cells? We will need the cells at this density, 1.2 millions of cells per milliliter at a viability of 95%. That means that 95% of the cells will be alive. Okay? So the first thing that we need is just preparing the culture. What is the second step? The second step is easy. It's just adding the DNA to the culture. This step is called triple co-transfection. We add the OSK DNA, the RepCap DNA, and the helper DNA. Here you have the 3 DNAs that are added to the cell culture. And we add these 3 DNAs with a detergent. The detergent will help the cells to absorb the DNA. It's called a cationic lipid. It's a positive detergent that interacts with the DNA that is negatively charged. Okay, so we add the DNA with the detergent and we incubate the culture in an incubation chamber. And the parameters that we set in the incubation chamber are 37 Celsius degrees, 8% of CO2, 80% of humidity, and 110 RPMs. This is the shaking force. Okay, so the incubation chamber will provide these conditions to the cells in order to produce many EV-OSK. Okay, so we incubate it, okay, at these conditions, and the cells start producing EV-OSK. And the day after, we enhance the expression of the EV-OSK by adding a booster. So 18 to 22 hours post-transfection, transfection means the addition of the DNA, we enhance the cells by adding sodium butyrate or another enhancer. There are many enhancers that you can use, and we keep on incubating at 37 Celsius degrees, 8% of CO2, 80% of humidity, and 110 RPMs. So this third step is just the addition of a booster. What is the fourth step? The fourth step is performed daily. It's just checking and maintaining the culture every day. We will check whether it is contaminated. That means that it smells not very good. We will check other parameters like the viability. That means how many cells are alive, and the number of cells. That is to say that they are not decreasing in number. So we count the cells daily and record the cell parameters, the cell density, the viability, and whether it is contaminated or not. If it is contaminated, we will have to stop the process, and yeah, that would be not nice, to be honest. Well, we continue incubating at 37 Celsius degrees, 8% of CO2, 80% of humidity, and 110 RPMs. This is a nice figure with the conditions. You see here, right? The cell culture incubating under these conditions in an incubation chamber. And what is the fifth and last step? Is harvesting. So it's collecting the EV-OSK. Is after the process you want the EV-OSK, right? So this is the fifth step. So it's harvesting EV-OSK that is secreting in the medium of the culture. So as we incubate the cells, they produce EV-OSK and they secreted the particles to the medium. So they expel the virus to the medium. So the virus with the DNA will be in the medium. And to recover the virus with the DNA inside, we centrifuge the culture. You see here, right? It spins. We centrifuge. We spin it in a centrifuge machine. When you centrifuge the cell culture, what you are doing is separating the medium and the cells. Okay, so we want the medium. So we keep the supernatant, that is the medium with the EV-OSK, at minus 80 Celsius degrees. So once we separate the medium, we just freeze it. and keep it in a freezer. Okay, and what do we do with the cell pellet? We just resuspend the pellet again so that it starts producing EVOs and releasing it to the medium. So what we do is add fresh expression medium to the cells and resuspend the cells so that they keep on producing and secreting the virus to the medium. After resuspending the cells again in fresh medium, we continue incubating under the conditions mentioned that are 37 Celsius degrees, 8% of CO2, 80% of humidity, and 110 RPM of shaking force. And this step is repeated 3 times at 72 hours, 96 hours, and 120 hours. So we will harvest the medium with the virus at these 3 times. Okay? So let's repeat the 5 steps. The first step is just preparing the cell culture. We will leave it ready to produce. We do it the day before. So the first day, what we do is adding the DNA with the detergent, okay, so that the DNA is absorbed by the cells. And we start incubating the culture with the DNA inside. And it will start producing EVOSK. And the day after, we add the booster. Okay, this is a little help. It will increase the production of the EVOSK. The fourth step is performed daily, which is my checking the culture and maintaining it. And the fifth and last step is just harvesting. It's separating the medium from the pellet. We keep the medium with the EVOSK, and we add fresh medium to the cells. We add fresh medium, we resuspend, and we keep on incubating for the next harvest. Okay, so happy, easy. Let's go to see step by step. Let's start with the first step. Let's go for it. Okay, here we are in the first step. Very easy. It's just preparing the culture the day before. Okay, we prepare the cells, the culture, the mammalian cells that are called HEK293. And the stages of this step are checking the pre-culture the day before. We will check around 5 liters of this culture. We'll check the smell, the color, and other things. We want the cells to be healthy. And these 5 liters will be diluted in 10 liters volume. Okay, so the 5 liters will be a little bit diluted, half diluted in 10 liters of culture. And after the dilution, we will incubate the cells under these conditions. During the incubation that will last 24 hours, the cells will double its population again. Okay, so the day after, they will be double. The day after, I have told you, we culture we check the culture again. We want it to be healthy, okay? So let's go for the steps within this step, the first step. Okay, first thing that we do is checking the 5 liters of culture. You see here, we check the smell. It has to smell good. We check the color. This is a nice color for this kind of cells. Yellow would be not nice, or I don't know, another kind of color would be not nice. So this is the color that these cells have to be. So we check the smell, we check the color, and we check the amount of cells that we have. So we check the density of cells using a cell counter. We will see it in following videos. Okay, so we check the cells. Okay, the smell is good, the color is good, the density of cells is good. The density is around 1.2 millions of cells, okay? And we need 95% of the cells to be alive, to be healthy. The cell counter will tell us. So what we have to do is diluting this culture, okay? We will dilute it. We will reduce the density. And yeah, because we want a final culture of 10 liters. Okay, so for this purpose, in order to dilute, we add more expression medium, pre-warm medium, and after adding the pre-warm medium, we swirl a little bit so that the cells distribute well across the culture. This medium is called Freestyle TM293 and is pre-warm. Before adding the medium, we have to pre-warm it at 37 Celsius degree so that the cells are happy. Okay, so let's have a look here. Do you see this picture? It's just the 5 liters of culture diluted with more medium, fresh medium. So we dilute the 5 liters in half. They will be now in 10 liters. And after diluting the 10 liters, we check the density, the density of the cells. It will be half because we have diluted it twice. So it will be around 0.6. Remember, the peak culture was at 1.2 millions cells per milliliter, and the dilution will be around 0.6 millions cells per milliliter. Okay, so once we have diluted it very well, we have swirled it a little bit and the cells are happy, we put the culture inside an incubation chamber. You see here, this is the culture inside the incubation chamber, and we set the incubation chamber at certain parameters. Okay, this is the display. Here we will set the chamber. And as you can see here, there are many chambers, so you can have a taste on how they look like. Okay, and what are the conditions for incubation? Okay, we will incubate 24 hours at 37 Celsius degrees, 8% of CO2, 80% of humidity, and 110 RPMs. This is the shaking force that the incubation chamber will exert. So as you can see here, the flash will be here with the cells happy, happy under these conditions. Okay, so we leave it this for 24 hours. And the day after, what do we have to do? Okay, so the day after, remember, we have these 10 liters of cells, okay, that were at 0.6 millions per milliliter. And these cells, you have to know that they increase their population, they double the population in 24 hours. So the day after, we expect to see this quantity, we expect to double this quantity. So we check the medium again. So we smell the medium, we check the color, okay, nice color, nice smell, and now we check the cells. And the cells will be around 1.2 millions of cells per milliliter. That is super important because this density is the optimum density for co-transfecting. That means for including the DNA. The other parameter that we check is the percentage of cells that are alive. It has to be around 95%. Okay, so the viability is 95%. So that is that step. Let's go for it. We have the 5 liters at 1.2 millions of cells per milliliter. We dilute it up to 10 liters. And after the dilution, what we have to do is incubating. So we incubate the cells under these conditions for 24 hours. So in 24 hours, they double again the density, and they are again at 1.2 millions of cells per milliliter. We make sure that the color is good, the smell is good, the density is good, 95% are alive, and we carry on with the following step. Okay, easy peasy. Let's go for the second step. Okie dokie. Step number 2, add the DNA to the culture. This process, adding the DNA to the culture, is called co-transfection. You see here in this picture, You see the 3 DNAs that are added to the culture. This is the culture, and these are the cells, okay? The mammalian cells. Well, it's very important to keep the ratios. Ratios are very important, okay? The ratio for the OS genes is two parts, two parts of OS DNA. 1.5 part of the RepCap and 1 part of the helper DNA. Keeping these ratios is very important, but no worries. I have calculated the amount of DNA that you have to add. I have done it for you. Okay? So let's read with me. In this slide, we will read together a lot. So the second step is add the DNA to the culture. It's called triple because we have 3 DNAs. Co-transfection. Transfection means adding the DNA to be absorbed by the cells. Okay? And the DNA is the host DNA, the RepCap DNA, and the helper DNA. Okay, steps. Very well summarized. Is mixing the DNA with lipids, cationic lipids. I have to mention that the DNA is negatively charged because of the phosphates, and we add a detergent. Okay, the detergent will dissolve the DNA, will encapsulate the DNA, and this detergent is positively charged. So the detergent that is positive will interact with the DNA that is negative. Okay, so it will encapsulate, and the detergent forms kind of small capsules that engulf the DNA, and these small capsules are able to be absorbed by the cells. So that is the principle behind the inclusion of the DNA inside of the mammalian cells. The mammalian cells need to absorb the DNA by using detergent. Okay, we call it cationic lipids. It is just detergent. So the first step, very well summarized, is mixing the cationic detergent, the positive detergent, with the DNA that is negatively charged. So we mix the detergent with the DNA, okay? And after mixing the DNA with the detergent, the only thing that we have to do is adding to the culture the mixture. Easy, right? And what is the third step? The third step is incubating, just incubating under these conditions that we are usually using. That is 37 Celsius degree, 8% of CO2, 80% of humidity, and 110 RPMs. This is basically what we are going to do. And I have broken down these steps so that you can see it more clearly. Okay, so let's go for the first step in here. Let's read with me. Okay, we have our culture, okay, that grew the last day. So we have it here, nice, the smell is good, the the color is good, and we count the cells. We count the cells and adjust the density to 1.2 millions per milliliter. And how do we adjust the density? Remember, by adding the medium, fresh medium, freestyle TM293. Okay, so in this step, we just uh, we just make sure that everything is all right, and we adjust again the density by adding the medium. Okay, remember we have 10 liters, 5 flasks of 2 liters. 5 times 2 is 10 liters in total. Okay, and once we have the correct density, we in the 10 liters we discard 0.5 liters. We discard this 0.5 liters because we are going to replace it with the mixture of detergent and DNA, so that at the end we will have a final volume of 10 liters. Okay, so in this step is just adjusting the cell density, and discarding a little bit of the culture. So we have the culture ready here. So we leave the culture there, incubating or whatever, and we go for the following step. The following step is mixing the DNA all together. The three DNAs, we are going to mix the three DNAs all together, respecting this ratio 2 to 1.5 to 1 part. Okay? And I have calculated the quantities. We mix 4.4 milliliters of RepCap that was at 1.5 milligrams per milliliter with we mix with 3.3 milliliters of DNA helper, which is 1.5 milligrams per milliliter. Okay, and we mix 2.2 milliliters of EVO's DNA, which is 1.5 mixed milliliter. So we mix the three DNAs together, and we add um, um, OptiPro. OptiPro is just for diluting. It's an agent in which the DNA is comfortable and in which the cells are comfortable. So we mix the four things together. Remember, 4.4 milliliters of RedCap, 3.3 milliliters of helper, 2.2 milliliters of EV-OSK, and 190 milliliters of OptiPro. We mix these four things together, the DNA and the OptiPro, in a bottle, and we leave it at room temperature. So we have here the DNA mixture. Okay? See, you can see here we are transferring. In this picture, you can see how we are transferring the DNA, the OptiPro, to a bottle. Okay, so now we put this DNA mix aside and we go for another step. Another step is preparing the detergent. In order to prepare the detergent, we just mix 30 milliliters of PEI. PEI is the detergent, the positive detergent that we are using. Okay, it's called PEI Max. PEI Max. Well, you have it here is 1 milligram per milliliter of PEI Max. That is the concentration. Okay, so in order to prepare the detergent, we mix 30 milliliters of PEI Max, the detergent, with OptiPro, with 170 milliliters of OptiPro. And in order to mix this properly, we swirl it gently. Okay, so here you are. We have the PEI Max, 30 milliliters, the OptiPro 170 milliliters, and we mix it together. And with the mixture is called PEI Max Mix. We call it the PEI Max Mix. Here you have a picture in which I am mixing the PEI Max with the OptiPro. Remember, the OptiPro is just something to dilute with. Okay, so now we have the three things that we need. Now we have the 10 liters of culture. We have the DNA mixture and we have the detergent. The next step is preparing the detergent and the DNA. Okay? Here you see the DNA mix. Okay. Here you see the PEI Max mix. So we mix both together. As you can see here in this step. Okay? Here we have the DNA mix and the PEI Max mix. That is to say, the DNA mix and the detergent mix. And we mix both together in a bottle. Okay? We mix it by swirling gently. Okay? Swirl it gently. And this step is very important. It has to incubate, this mixture has to incubate 15 minutes at room temperature so that these small capsules are formed. In each capsule, you will find the detergent in the surface and the DNA inside. Okay, they are kind of spheres with the detergent outside and the DNA inside. So we leave the 15 minutes at room temperature the mixture, and the mixture will start forming these spheres that I am telling you. Okay, so once we have the mix, it's important to respect these 15 minutes, no more than 15 minutes and no less. What we do is adding the final mix. This is the final mix, okay? The DNA plus the detergent. So we add the final mix to the culture. Here you have, this is a picture in which I am adding the mix, the final mix, to the culture. Okay? The final mix added to the culture. And once the cells see the mix, they will recognize these spheres that have the detergent outside and the DNA inside. So the membrane of the cell will start engulfing, will start including the detergent with the DNA. The detergent is the agent that makes it possible. Okay? So we add as I have told you, we add the final mix to the cells. Remember, there are 10 liters of cultures divided in five flasks, each flask having 2 liters. Okay, so we have the 5 flasks with 2 liters each, total of 10 liters. And the only thing that we have to do is, oh yeah, the last step, leaving it incubating. Okay, because we need our cells happy incubating and the conditions that they like, and I'm going to tell you. So during the incubation, the cells will accept the the DNA inside thanks to the detergent, and once the DNA is inside, the DNA will do the magic. Okay, but we have to leave it incubating. Let's repeat the incubation conditions. The incubation conditions, as always, are 37 Celsius degrees, 8% of CO2, 80% of humidity, and 110 RPMs. So we leave them inside the incubation chambers. Here you see the inside of the incubation chamber, and on the top you see the display with the conditions. We set the conditions here. Okay, so we incubate it. Now we are ready to go for the following step. And before going to the following step, let's summarize it in order for you and me to have it clear in our minds. So the first thing that we did was preparing the cells. The second thing that we did was mixing the DNA. So, so at this point, we had the DNA prepared. Then we prepared the detergent. Okay, after preparing the detergent and the DNA, we mix the detergent and the DNA. We mix it here together. And once we have the final mix, which is the detergent and the DNA, we add the final mix to the culture. And once it is added to the culture, we just incubate it, leave it incubating, and the magic will happen now. In the incubation, the cells will start producing EVOs inside. So here is where the magic happens. Okay, so let's go for the next step, the number 3. Well, step number 3, boost the expression 18 to 22 hours after transfecting. That means help the expression by the addition of a booster. Very easy step. Okay, so boost the expression 18 to 22 hours after transfecting. Okay, after 18 to 22 hours after the, the transfection, we enhance the cells, we enhance the expression by adding sodium butyrate or another booster, another enhancer. They are called boosters or enhancers. Okay, so this step is very easy. We just add the booster and keep on incubating. Okay, as easy as that. Just this, adding sodium butyrate. Okay, in this step, the stages are 3, as you can see here. So we have the cells in the incubator, the culture, we take the, the culture out, and the first thing, okay, we check the cells. We check, as always, the smell, the color, the density, and the viability. We have to say, well, you can see here, right? The cells start producing a little bit of EV-OSK. Just a little bit. Remember, it's only has been 22 hours. So EVOs hasn't been produced. We haven't produced much EVOs, okay? And the growth of the cells decreases as the production of EVOs increases. So we will see as the cells start decreasing, not decreasing, but the growth decreases. So it will not double anymore in number. In fact, it will be steady. Okay, so we check the cells, the color, the smell, the density is all right, and not many of them have died. And what we do is collecting the sodium butyrate. Okay, so we take the sodium butyrate and we transfer 100 milliliters of sodium butyrate to the culture, just like this. We can use a pipette boy, as you can see here, to transfer the 100 milliliters. So this is basically the step, just adding 100 milliliters of sodium butyrate. Okay, once we add the sodium butyrate, we return the culture to the incubation chamber. And remember, we will incubate at 37 Celsius degree, 8% of CO2, 80% of humidity, and 110 RPMs. As you can see here, again, the same picture with the culture inside the incubator, and it will incubate. Well, we have to say that the booster, the cells will produce much more EVOs, as you can see here. It will start producing EVOs like crazy, a lot of it. So that's it, folks. This is the step number 3. Let's go for the number 4, which is very, very easy as well. Let's go for it. Okay, step number 4, as I have told you, is a very easy step. Okay, so let's go for it. The number 4 is checking and maintaining the culture in a daily basis. Checking and maintaining the culture daily. Okay, so the only thing that we do is counting the cells daily and record the cells parameters, the cells density and variability. And as always, we check the smell, the color, and what I have mentioned. And after checking, is just continue incubating the culture, because we want the cells to produce a lot of EVOs, right? Okay, let's have a look here in the steps. You have your culture. Well, we know this picture. We have seen a similar picture a lot of times, right? So we just take the culture out of the incubator and we check the smell, we check the color. The smell and the color will tell us whether the culture is infected or not. So this color is a nice color, and the smell has to be a nice smell. And we check the density and the viability. You see here, after several days, the cells start producing a lot of EVOs. So, well, it produced and secreted to the medium a lot of EVOs. You see here, we have a lot of EVOs. Happy for that. Okay, so we have our 10 liters of culture producing a lot of EVOs. I have included a couple of pictures here. As you can see here, this is a cell counter. We just put the cells here with a dye that colors the cells in blue. Okay, and we introduce it here in this slot. And it's very, very easy to operate. The only thing that we have to do is pressing a button, pressing start. And what you will see will be something like this. the cell counter counting the cells. The display will show something like this. You see here, it will indicate you the density of the cells. Well, this is not the counting of this culture, okay? So it's just an example. So don't be misled by these figures, but you see here. The density will show you the density and also the amount of cells that are alive. In this example, for example, 68%. But that would not be a nice percentage of cells alive, okay? But I wanted to show you how it works. It's just including here the sample, introducing here in the slot, pressing start, and it will count the cells, the density, and also the amount of cells that are alive. Well, once we check the smell is good, the color is good, it's just returning the culture to the incubator, to the incubator. The 10 liters, we will return the 10 liters to the incubator, and it will keep on incubating at as always at 37 Celsius degree, 8% of CO2, 80% of humidity, and 110 RPMs of shaking force. And it will continue carry on producing a lot of EVOs, a lot. Okay, so let's go for the last step, the fifth one, and last, very easy as well. Okay, it's time to harvest. Is the fifth and last step. Step number 5, harvest EVOs from the supernatant. And yeah, the key point here is just centrifuging. By centrifuging, you see here, by centrifuging the culture, we will separate the supernatant, that is the medium, and the pellet. Okay, the pellet with cells, and the supernatant, the medium with OSK, EV-OSK. Okay, you see here, right? And the small EV-OSK particles, and in the pellet here, in the bottom, the cells. Okay, so let's go for it. Step number 5, harvest EV-OSK that is secreted by the cells in the medium of the culture. Okay, so we centrifuge the culture 72 hours after transfecting to separate the medium and the cells. As you can see here, the cells will be in the bottom of the tube, and the supernatant with the EVOs will be on top of the cells. Okay, so we centrifuge and we keep the supernatant with EVOs at minus 80 Celsius degree. So we will freeze the supernatant. We add fresh medium to the cells. We add new fresh medium and resuspend the cells so that they keep on producing and secreting EVOs in the medium. That's it. So once we harvest the supernatant, we add more medium. We resuspend, and we keep on producing more supernatant with EVOs. Okay, so we continue incubating, and we repeat the process several times. We want to produce as much supernatant as possible, so we repeat this step 5 a couple of times more. So let's read with me. Let's see the steps here. First thing that we do is centrifuging. Okay, you have a representation here of the culture being centrifuged, and you see here the culture were distributed into bottles. The bottles were centrifuged, and here, as you can see, the pellet will be here, and the supernatant, the beautiful supernatant, here. Okay, once we have the pellet and the supernatant separated, we pour the supernatant into bottles. You see here, bottles with supernatant and with beautiful EVOs. Yeah, these bottles will be frozen. The final product is here. Oh yeah, EVOs, what we want. That's why we are so happy, and we put smiling faces and hearts. Okay, this is what we want, the supernatant with EVOs. Okay, and regarding the cells, the pellet cells, we can use them again to produce more supernatant with EVOs. So what we do is we take the bottle here, put it here, and we add more fresh medium. So we add fresh medium to the pellet and we resuspend the cells gently in the medium. In order to resuspend the cells, that's it, to distribute the cells again in the medium, you can use a spatula or a pipette like here, and we stir the spatula until all the cells get well distributed among the culture, among the medium. Okay, we return the resuspend pellet to a sterile flask. That's it. We put the culture again in flask, and we put the flask again in an incubation chamber to incubate and to keep on producing more supernatant with EVOs. Okay, understood? Easy? So that's all, folks. That's all. After getting this and doing all the processes, we will repeat these 1, 2, 3, 4, 5 steps after 96 hours. So we will repeat this step after 96 hours so that we will get more supernatant and we will keep on producing more. And after 5 days, we just repeat these 2 steps. After 5 days, all the process will stop. We will just freeze the supernatant and we will not use anymore the cell pellet. This is the final product. This is the EV-OSK. Okay, so on producing EV-OSK, the only thing that is missing is just cleaning this. Cleaning this will be in the following video. Okay, so let's summary all the things that we have seen in this video. Let's go for the next slide, shall we? Let's go for it. Okay, let's have a look on the summary. Wow! Oh yeah, the five steps. Very easy. You remember, right? The first step was preparing the cells, the mammalian cells, the HEK293, the day before. Okay, we prepared the culture. And what was the second step? It was adding the DNA to the culture. That is called triple co-transfection. We add OS DNA, RepCap DNA, and the helper DNA. Here you have, right? Do you remember the DNA? We add to the culture. Remember with the transfection agent, with the detergent. Okay? And what was the third step? Enhancing the expression of EVOs by adding a booster the day after. So 18 to 22 hours after transfecting, we just add the booster. Okay? And it will boost the expression. We will have a lot of EVOs by boosting it. Okay? What was the fourth step? Checking and maintaining the culture every day. This is the fourth step. Okay, while we take the cells out of the incubator and we check them. We check the color, the smell, the density, in other words, if it's growing or decreases in number or whatsoever. Okay, it's just a checking. And what is the fifth and last step? Is harvesting. Once the EVOS is produced, is secreted to the medium by the cells. In the when it's in the medium, it's just the harvesting. And remember, what we do to harvest is just centrifuging. So by centrifuging, we separate the pellet with the cells and the supernatant that is on top with plenty of EVOs. Okay, so it's just taking this, freezing it, and oh yeah, there we are, there we are, there we have what we want, what we need. So that's all, my friends, for this video. I hope you have enjoyed the video. And remember, you will find this information well detailed in novosumer.com in the following months. Okay, this information, this presentation will be available along with a proper protocol with a proper working instruction, SOP, as you want to call it. And the name will be something like this. So you can look for it on the website, and it will have a name similar to it. Large-scale expression of AAV-OSK virus in HEK293. And remember, all these protocols are compliant to CGMP, ISO 9001, ISO 45001, ISO 14001. In other words, they are compliant to standards of quality, standards of health and safety, standards of environment, of respect for the environment. Okay, so you will be fully able to implement it in case you are an entrepreneur and you want to develop this technology in your industry, in your company. You will be able to have all the information that you need. These protocols include materials and method descriptions, well-detailed descriptions on how to perform the steps, everything, literally everything. Okay, so there are only a few words to say. Novosumer is here to say big thank you very much. Again, I am the PhD and scientist Eric Amado. You can find me in novosumer.com or either in the with this address, email address that you see here. Okay, so my friend, just be happy, have a beautiful day, and enjoy. Okay? Enjoy. Thank you very much. Bye bye.